Hey, do you want to know what goes on in your muscles during a leg extension? Keep watching to find out through the sliding muscle filament theory.
It all starts with the stimulation of the nerve fibers. During the resting and polarized state, the surface membrane of the muscle cell is permeable to potassium ions and impermeable to sodium ions. This causes there to be more sodium on the outside and more potassium on the inside. When the stimulus is great enough, motor units are recruited. Motor units are places where nerves will stimulate areas of muscles to work. From the central nervous system, we can utilize all sorts of sensory information to help us determine what type of action will occur. First, the stimulus needs to be great enough to create a response, which means exceeding this threshold, altering the permeability of the membrane, allowing for sodium in and potassium out. When the nerve fiber is stimulated, the resting potential is gone and depolarization occurs. Depolarization is when the membrane permeability is altered and the inside goes from being more negatively charged than the outside. And after depolarization, repolarization occurs quickly afterward because the membrane is now only permeable to potassium ions, as the inside of the membrane is negatively charged, bringing the sarcolemma back to normal. The muscle fiber is ready for more stimulation.
Now let's take a look at our muscles. All throughout our bodies, we have muscles that help us move. These are called skeletal muscles. These skeletal muscles are striated and are attached to two tendons. Within these skeletal muscles, there are various bundles of fibers. These bundle of fibers contain cylindrical, striated cells or muscle fibers, and then these muscle fibers contain myofibril. Within these myofibrils, there are two filaments, the actin filament and the myosin filament. These filaments make up the smallest unit of muscle, which is called the sarcomere.
Around these different parts of muscles are the epimysium, wrapping around the bundles of muscles, and then the perimysium, wrapping around the muscle fibers, and the endomysium, wrapping around the myofibrils.
To review, the breakdown of the muscle goes from the skeletal body to the bundle of muscles, and from the bundle of muscles to the muscle fibers, and from the muscle fibers to the myofibrils, containing the actin and myosin filaments, which create the sarcomere. Here is a sarcomere and its components. The A band is the length of the myosin, the H zone is in the middle of the sarcomere, the I zone is the distance between myosin to myosin. The Z lines make up the outside edge of the sarcomere and attach to one another in a line. The actin filaments are attached to the Z lines while the myosin is not, as it only floats in between. The myosin has long strands, a neck that attaches two globular heads, and a neck that flexes towards its tail, pulling in the actin and Z lines. The length of the A band and the H band shorten while the I band stays the same, which causes the muscle to contract by 1/3, which is 2/3 of its resting length.
Around every myosin, there is going to be 6 actin, and around every actin, there will be 3 myosin.
To review, the sarcomere is composed of the two Z lines with actin filaments attached to it, myosin filaments with globular heads attached to it, the H zone, which is in the middle of the sarcomere, the A band, which is the length of the myosin, and the I zone, which is the distance between myosin to myosin. During contraction, the H and A zone shortens while the I zone stays the same, as the muscle contracts by 1/3, which is 2/3 of its resting length. Well, how does the contraction of the muscle even begin? Well, all muscle contraction begins with calcium, which is stored in our bones. And this is tropomyosin, a double long-stranded protein, which is aligned nearly parallel to the actin filaments. And this is troponin, a protein complex of 3 globular heads along the actin filament. One globular head attaches to the tropomyosin, another to actin, and the final one binds with calcium ions. Globular heads attached to the myosin have to go through tropomyosin to attach to the binding site on actin, but in a resting state, troponin won't let them through. If there is no calcium, troponin will not let tropomyosin off the binding site, and the globular head goes nowhere. The globular head wants to attach to the binding site. When calcium is released into the sarcomere, the calcium is going to bind with the troponin. Then troponin is going to pull tropomyosin off the binding site. Once the binding site is open, the globular head will bind and contract the muscle. During the state of relaxation, the myosin binding site, where the attachment occurs, is blocked by the tropomyosin, not letting myosin cross bridges to attach the actin. Calcium ions then bind to each troponin when it's released into the sarcomere, and the globular head brings tropomyosin out of the way so that myosin can bind to actin. During contractile stroke, myosin is energized with ATP, causing the head to tilt and propelling the actin. To review, when calcium is released into the sarcomere, the calcium is going to bind with troponin. Then the troponin pulls tropomyosin off the binding site. Then the globular head will bind with actin and contract the muscle. And during relaxation, contraction does not occur because there is no calcium to bind with troponin to pull tropomyosin off the binding site.
When we need muscles to contract, we send a message along the efferent pathway where sodium in and potassium out. And when it gets sent down, it's going to travel along the axon to the neuromuscular junction, which is the connection point between the nerve and the muscle. Acetylcholine, ACH, are neurotransmitters that gets released from the nerve side, and they get released into the synapse, which is the gaps between the nerve and the muscle. It gets received by the receptors, causing muscles to contract. These chemicals received by the receptors are going to stimulate more action potentials, and more motor units are going to be stimulated based on the sensory information. A wave of positive energy travels along the axon into the neuromuscular junction, where acetylcholine is released. This neurotransmitter is released into the receptors of the neuromuscular junction. Once that happens, it will continue to send the action potential along the sarcolemma, which is the wall of the membrane. Then it's going to travel down the T-tubules, stimulating IP3. And when it stimulates IP3, it's going to release calcium into the sarcomere from the sarcoplasmic reticulum. In the sarcoplasmic reticulum, calcium ions are trapped within, unable to bind to troponin, and no muscle contraction occurs. Once this gets released into the sarcomere, it's going to bind with the troponin, pulling tropomyosin off the binding site, allowing for the globular heads to attach to actin, and muscle contraction occurs. The calcium-driven pump brings calcium back into the sarcoplasmic reticulum, which prepares for the next contraction. To review, during muscle contraction, an action potential gets sent down the axon into the neuromuscular junction. Then acetylcholine gets released into the receptors and travels along the sarcolemma, down the T-tubules, stimulating IP3 to release calcium from the sarcoplasmic reticulum. Then these calcium ions bind to troponin, exposing the binding sites for myosin for contraction. And then ATP drives the calcium pump, bringing calcium back into the sarcoplasmic reticulum for the next contraction.
When your muscle is relaxed, the cross bridge is detached from actin. The cross bridge is a high energy state, which results from the previous binding and splitting of ATP, which stands for adenosine triphosphate. Adenosine triphosphate is basically stored energy, and it's made up of adenosine diphosphate and an inorganic phosphate. These products prepare itself for energization when they combine again while surrounding the myosin head. Its attachment occurs when the cross bridge with the floating ADP and P attaches to actin. When the actin and myosin interact, a release of energy goes to the globular head, tilting it and propelling the actin, which releases the ADP and P. This is called the power stroke. When the cross bridge is stuck to actin until further energy stimulation, we are in a state of rigor because no ATP is available, which causes the stiffness of the muscle. Then, when a cross bridge binds with ATP again, it causes the release of the cross bridge from the actin to be in the state of relaxation.
To review, the cycle begins with ATP charging the globular head in a relaxed state. Then the globular head attaches to actin, and then leftover energy flexes the neck of the head, and then it enters a state of rigor when the cross bridge is stuck to the actin. And then ATP binds to the globular head to release it from actin. Here's a summary of everything we learned in this video. In this video, we learned about nerve impulse, muscle structure, the composition of a sarcomere, how muscle contraction occurs, and the role of ATP in muscle contraction. And that is all for the sliding muscle filament theory.